Okay, we're going to continue and finish this up with our last and third of our urinary system PowerPoint videos. And this is just a little bit more of the fine-tuning about what really happens and how we regulate the urine volume. In our first video, we talked mostly about the anatomical structures. In the second video, we talked about the how urine is made. And that urine is made is basically a summation of filtration, which a lot of things are filtered, most of it's reabsorbed, and then there is some selective reabsorption as you go along the tubule, as well as secretion putting things back into the tubules. Hopefully you understand that. If you don't understand it, I recommend that you go back and review and go over the urinary system 2, again video 2, because that's going to help you a lot to understand what's really going on here, because there's a lot of little physiological processes that we need to get straight before we actually get any further. Okay, just about urine. We filter a lot. You know, the glomeruli are busy all day long and all night. They filter about 100 to 180 liters per day. It's about 48 gallons that they filter of filtrate. That's the stuff that gets through the glomerulus and leaks out into Bowman's capsule. You know, I mean, a kidney's this size, size of my fist. Well, what happens is most of that 100 to 180 liters of filtrate that I have per day is actually reabsorbed. It's pulled back in. So I finally end up with putting out about 1 to 2 liters of urine per day per person, okay, in a normal healthy kidney with normal dietary intake with fluid and stuff like that. And one word that's going to pop up that you might see is called glomerular filtration rate. Glomerular filtration rate. And glomerular filtration rate is just by definition the amount of urine that's filtered per minute, the amount of urine that's filtered per minute, because again, where does filtration occur? Occurs in the glomerulus. This becomes an important thing that they are able to actually measure and give a value for, okay, down the road. They could actually calculate the glomerular filtration rate in a couple different ways, and they're able to do this to be able to determine how effective the kidney is. As we see people going into kidney failure, what happens is the GFR, the glomerular filtration rate, continues to go down as the glomerulus stops filtering, okay, and that's what happens with kidney failure. So they're able to actually monitor and evaluate in regards to the level or degree of kidney failure someone's in by looking at the glomerular filtration rate, helped out by a couple of the tests besides that. So let's just talk about some of those things. Now, how does the body normally regulate the glomerular filtration rate? Okay, there are a couple different ways, 3 different ways mostly. One's called autoregulation. It has sort of like a gatekeeper itself that sort of keeps an eye on what's going on and is able to help out and make some minute-to-minute changes, because obviously things change as time goes on through the course of the day. Things aren't always stable, so we have to have a really fast and easy way to make some quick, easy changes, and I'll explain what those are. We also have hormonal regulation, and hormonal regulation, we talked a little bit before about something called angiotensin 2, okay, and we'll talk a little bit more about that. There's also something called an atrial natriuretic peptide, okay, or some ANP, which I'm going to say rather than atrial natriuretic peptide, which I have difficulty always saying. There's also something called BNP, and BNP is basically about the same thing, okay? So ANP and BNP, if you hear ANP or BNP, they both mean the same thing. And then we also have neural regulation, and we'll talk a little about neural regulation and what we do with that. So hormonal regulation with autoregulation, self-watching itself, self-monitoring itself in the area of the glomerulus. Hormonal regulation primarily involving angiotensin 2 and ANP, let me just go that way, or BNP. Also another hormone that I want to add in there that you might want to think about that we'll actually talk a little bit about, okay, is called ADH, Antidiuretic Hormone. And we'll talk a little bit about that as well in this particular talk. And then we have neuroregulation involving sympathetic activity to the area of the glomerulus and the nephron. Okay, let's talk about those individually. Let's first talk about this autoregulation, what really happens, okay? Now, the picture you have on the upper right shows that image that we've seen before. We see the afferent arteriole coming in at the top, okay, of the area of the glomerulus. You see the glomerulus inside that little capsule or Bowman's capsule that's collecting the filtrate, and you see the beginning of the proximal convoluted tubule. You also see the area of the efferent arteriole coming out at the bottom of that glomerular capillary, because we know that we go from afferent arteriole to glomerular capillaries to efferent arterioles to peritubular capillaries to venule. But what we also see right here is, if you look, there's another little loop that's right where the afferent and the efferent arterioles come close together, okay? And that's the area of this ascending loop of Henle right by where it starts to convert from the ascending loop of Henle into the distal convoluted tubule. Now you should know that what that is, because if you don't, go back to the previous video and you'll be able to see that easily. Right in there, there are some special cells, okay? And these special cells is in the area of what's called the macula densa and the juxtaglomerular apparatus. What happens is when the glomerular filtration rate is low, okay? So in other words, if I have very low blood that's going through the glomerulus, what happens is the glomerulus is filtering very little sodium and very little water. Okay, so we start to send start to filtrate going down the proximal convoluted tubule with very little water and very little sodium. Okay, well that finally goes from the proximal convoluted tubule to the loop of Henle, loops down and comes up and it gets to the top of that ascending loop of Henle, right where it's going into the distal convoluted tubule. That's this area right there. What happens is these little cells in that juxtaglomerular apparatus are able to detect the level of sodium and water in that area of the ascending loop of Henle, right where it goes into the distal convoluted tubule. And what they do is if it's low, it just means that there's just not enough blood flow going into the glomerulus. So what it does is it actually causes or actually releases any level of vasoconstriction on the afferent arteriole. And what that does is by the afferent arteriole, with the decreased vasoconstriction, allows the afferent arteriole to open and doesn't do much with the efferent arteriole at this time. Keeps the efferent arteriole where it's supposed to be. But by opening the afferent arteriole, and we talked about in the previous lecture, that what happens is the efferent arteriole is always a little bit smaller than the afferent arteriole in regards to diameter. What happens is now if I have efferent arteriole being a little bit narrower and the afferent arteriole being a little bit more open, I get more blood going into the glomerular capillaries as well as more back pressure because there's less waste to come out. So the glomerular capillaries get filled, they get engorged more, and they filter more. So basically what they do is by that afferent arteriole dilating, it will raise the glomerular filtration rate. They actually cause more filtration. Let me just explain that again. If I have less sodium and water in the area of the ascending loop of Henle, right by the juxtaglomerular apparatus and the sensor cells with the macula densa, what happens is then the afferent arteriole will relax. The vasoconstrictor activity on the afferent arteriole relax. Afferent arteriole, with the blood coming into the glomerular capillaries, increases in diameter, causes more blood flow into the glomerulus. As a result, if the efferent arteriole doesn't change, what happens is there's more backflow because again the efferent arteriole is smaller than the afferent arteriole. Now the afferent arteriole is even bigger, more blood going into the glomerulus. Therefore, there's increased pressure, hydrostatic pressure within the glomerular capillaries, more filtration. Makes sense, okay? And you see that as well on the bottom in the image I included on the bottom. They just have it backwards, flip the other way. One's right hand, one's left hand. You see where the afferent arteriole goes in at the top, and then as it goes into the glomerular capillaries, which we see down in here, you see this area right here is the juxtaglomerular cells, okay, measured by the macula densa, which is right here. So here's my ascending loop of Henle going into the distal tubules right at the beginning. It's right at the junction of it. So some people will say ascending loop of Henle, some people will say distal convoluted tubule. I don't really care. It's right there, okay? And what happens is it'll measure the amount of sodium and water in this area right here, and if it's low, it means that there's been low filtration. Therefore, the afferent arteriole will widen up, it'll open up, therefore increases the blood flow into there, more restriction come in here, and as a result, the pressure inside the glomerular capillaries increases, and will increase my filtration, which means that then now I'll increase the level of water and sodium in the proximal convoluted tubule, which gets the ascending loop of Henle, blah, 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 and on and on. And basically that's what we see with that, okay? We also have hormonal control. And the first place about the hormonal control, we talked about this a little bit in the last lecture as well. Again, if I have low glomerular filtration rate with low fluid and salt in the juxtaglomerular apparatus, what also happens if it's sufficient enough, there are little cells there that release a certain hormone called renin, okay? And the juxtaglomerular apparatus with the renin converts a precursor, angiotensinogen, to angiotensin 1. Okay, angiotensin 1 then gets into the bloodstream and it goes to the lungs. Well, that goes to the lungs. I don't know who figured that one out, but goes to the lungs, and in the lungs there's an enzyme called angiotensin converting enzyme, ACE, ACE. And what happens is that angiotensin 1 is converted to angiotensin 2. Angiotensin 2 is a very potent vasoconstrictor. So what happens is now I could increase the amount of glomerular filtration even more, where in the first place with the autoregulation I'm dilating the afferent arteriole. In this case, I might have that afferent arteriole dilated but I need more. Now I trigger this and the efferent arteriole constricts more. So now there's a greater difference between the size of the afferent arteriole, much larger than a much smaller efferent arteriole, backs up the pressure in the glomerular capillaries, and therefore it improves it, or it increases the filtration. Also, what happens is one further step is besides causing vasoconstriction of the efferent arteriole, the angiotensin 2 goes to the adrenal gland, which is on the top of the kidney, so it's really close. And in the adrenal gland, it stimulates the release of another hormone called aldosterone. Aldosterone then goes back to the kidney, okay? And if you remember, we talked about in the ascending loop of Henle, there was an area where the wall of the ascending loop of Henle kept water in, okay? And what happens is the ADH will actually allow more water out there as well as in the collecting duct. It sort of makes it more porous and let more water out, so actually will results in water retention. So when you start to retain the water instead of keeping it in the tubules, which eventually ends up in the kidney and eventually ends up in the toilet, we save that water. When I have increased water that's saved in the body, I put more of that water into the vascular structure, which then increases the volume intravascularly, which raises blood pressure as well. So we have a couple different mechanisms whereby this angiotensin does work. Okay, and that's one way we have a hormone control. And again, when we talk about how we concentrate and dilute urine, basically that's that antidiuretic hormone. Okay, let me just mention a little bit more about the antidiuretic hormone, which I was talking about already. And the ADH is actually a hormone from the pituitary. Okay, the pituitary in the brain. Okay, little thick stalk that we talked about that hangs down from the bottom of the hypothalamus. And what happens is we're able to, the body wants to conserve water, okay? And when the body wants to conserve water, if we're dehydrated or something like that, we want to make lose less water. So in other words, if I've sweat a lot, lost a lot of fluids, vomited and stuff like that, and all of a sudden I'm decreasing my intravascular volume, which will decrease my blood pressure, I could conserve water because the pituitary gland with the ADH from actually it's the back half of the pituitary gland sends out this ADH, and it goes to the area right at the end of the ascending loop of Henle and at the distal convoluted tubule and at the collecting duct, and it said, instead of keeping the water inside the tubules, let's make that tubule a little bit more porous and suck that water right out and put it back in the circulation. So it takes the water out of the tubules and puts it back in the blood, okay? And that's why it's called antidiuretic. Antidiuretic. Anti means against, diuresis means urination. So in other words, antidiuretic means urinate less, okay? So the amount of water is absorbed, okay, and depends upon a lot of things, osmotic gradient in the cortex, or the medulla, excuse me. As I get deep in the medulla, things get more concentrated. We talked about that before. But let's just get back to that and saying what happens is when the ADH is secreted by the pituitary, it affects how much water I take out of the renal tubules in the end of the ascending loop of Henle, the distal convoluted tubule, and the collecting duct. It takes more water out. It conserves more water. It saves more water, removes it from the tubules, puts it back in the blood. That increases intravascular volume and raises the blood pressure. So we could actually keeps and monitors and maintains body fluid levels, which we need, because we know we're mostly fluid, okay? So that's what the ADH is really important. So it helps to prevent excessive amounts of urination. There's a thing, another condition is called diabetes insipidus. You hear about, everybody knows about diabetes mellitus, okay? But diabetes insipidus is basically a situation where you lose that ADH control, and people will lose large volumes of fluid, excessive amounts of urination, and it becomes a problem. And it's a problem with ADH. ADH doesn't work anymore in those individuals. What also affects ADH? Well, some simple things. Alcohol and caffeine. Do you ever notice when you drink a lot of coffee, you have to go to the bathroom a lot? Because the caffeine actually blocks the effects of the ADH. Alcohol does the same thing, okay? It makes the walls more impermeable to water as compared to more permeable. ADH makes them more permeable. Alcohol and caffeine makes them less permeable. So therefore, what happens, they can't get the urine out of the tubules. It stays in the tubules. They expel and they excrete larger volumes of urine. They lose more fluid, okay? And that's where the ADH comes in. It's sort of an important type of a hormone, and that's just a little flow chart that shows you what happens that you could look at on your own spare time after you listen to what I've said and looked at it, okay? What happens is, I should mention this, and I'm going to bring this in. I talked about this before, and this is called renal hypertension, okay? And the picture that we have in the upper right is basically a renal arteriogram. The large black line down the middle, okay? This area right here, let me do it. This area in here, okay? This area here, 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 here. Guess what that is? That's the aorta. Okay, everybody figure that out. Aorta. And you can see the renal artery. Here's a renal artery coming out here. Here's a renal artery coming out here. Now let me get rid of that, what I've drawn in. And what you see, if I look at the renal artery there, there's an area right there that's narrowed, and there's an area right there. How do I know? The arrow is pointing to it. But actually, you can see how the dye that they put in the artery, in the aorta, is sort of like limited right in this area and right in this area. Okay. Well, what happens is that decreases the blood flow. So when that blood flow is decreased, we have decreased filtration. That goes back to do everything we've talked about before. It's detected in the macula densa, the glomerular apparatus gets activated, and what happens is we get that renin and taken to the angiotensinogen to angiotensin 1 to angiotensin 2, and that increases blood pressure. The problem with this is some people have a form of hypertension which is called renal hypertension. And in this situation, when you try to treat these people with medications, they don't respond. Why? Because the medications don't change the narrowing of the renal arteries, and the problem continues to exist. So you can't control it very well. These people actually, what they'll go in is they could go in and they may balloon it open, just like they do with a coronary artery. Like we talked about how they could take a catheter with a balloon on the end of it, stick it in the renal artery, and blow it up and put a stent in there, and that will open up that renal artery. Guess what happens to their blood pressure? It goes down. Now everything is fine because now the renal blood flow seems to be okay. Okay, and again, this also talks about how the angiotensin 2 stimulates the adrenal gland to secrete aldosterone, causes the nephron to retain sodium, and therefore, one other thing I want you to remember, okay, and this is sort of important in many places, where sodium goes, water goes. In other words, if sodium is retained, water is retained. There's very little time when they're separated. Okay, in fact, it's funny because when I look at the blood levels of sodium, if I took a little bit of blood from a vein and looked at the serum sodium level. Serum means the sodium that's in the plasma. If I look at the serum sodium level, guess what the serum sodium level shows me? In most cases, how much water there is. It looks at it sort of like an obtuse way. If my serum sodium level is high, okay, it means I usually have too little body water. There's something that's causing a loss of fluid, but the sodium gets retained. If my serum sodium level is too low, okay, is lower than normal, it usually means that there's too much water. It's dilute. Okay, so if it's too high, it's concentrated because there's less water, and if it's too low, it means that it's too dilute. There's too much water and less sodium, because usually they're in balance, but there's some diseases that will cause a change in that balance, and that's what we see. That's why we look at the serum sodium levels. Okay, so that's a little bit about that. Hormonal control, another hormonal control, this is the ANP or BNP, okay? And this is sort of a very interesting way, and I'll tell you why this test becomes exceptionally important. When the blood volume increases, okay, when blood volume increases, it actually goes to the area of the atria in the heart and stretches the atrial walls too much. We know those atrial walls are stretchable. Well, I take those atrial walls and stretch them a whole big long way. What happens, that stimulates the release of this ANP or BNP, okay? Same thing, okay? And what happens is what the ANP does, or BNP does, it promotes water excretion, which means it increases urination or diuresis. okay? And it just goes and says, hey, we're too full, we're expanding too much, let's get rid of some of this water. It goes to the kidney and causes water to be excreted. And so, and also, because water goes, it also does that with sodium. So sodium goes as well as water, okay? And as a result, now, because the atria was stretched, stimulated the release of this ANP or BNP, it caused the kidneys to excrete more sodium and water, volume goes down, and therefore the atria should go back and the stretch should be decreased. Makes sense, and the volume should decrease. Where this doesn't become usually the case is in heart failure, okay? And ANP or BNP is a commonly used test to evaluate the level of heart failure. Why? What is heart failure? Heart failure is exactly what it sounds like. The heart fails. What happens in heart failure, we have a situation where the ventricular muscle is not strong enough or is not working well enough to pump out what comes to it. Okay, so just think about that. The blood's coming to the ventricle, and instead of getting like a 60% ejection like we talked about in the cardiovascular discussions earlier, maybe you get 30% because the heart's just, I'm just going along, I'm tired, I can't do this, I'm weak, blah, 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 it's the end of the semester, I can't do, and things get not as strong. As a result, what happens if I get blood, the same amount of blood coming back, but I pump out less, what happens? The blood backs up. Where's the blood going to back up? Well, from the ventricle, it backs up in the atria. So in other words, I have less atrial contraction force to fill the ventricle, and now the atria, what happens to the atria? The atria starts to balloon and gets bigger. So in heart failure, one way that they follow it is they watch the ANP or BNP. If the ANP and BNP keeps on rising as a chemical level in the blood, they know the heart's continuing to fail. Why? Because the atria continues to stretch because the ventricle's not pumping out what comes to it. Very simple concept, easy to think about. Sometimes you have to, I know, I mean, sometimes it's the first time you're hearing these things, but and sometimes you have to hear about them 3 or 4 or 5 or maybe 10 times. But after a while, when you think about it, it says, my gosh, you know what? That makes so much darn sense, and it does in most cases, okay? So that's ANP or BNP, which is another way we control a glomerular filtration rate, okay? We also have neural control, okay? And again, neural control largely has to do with the sympathetic nervous system. What happens is if I want to raise the glomerular filtration, what do? I decrease my sympathetic stimulation. What does the sympathetic stimulation do to arterioles? It constricts them, causes a vasoconstriction, okay? Arterioles, vasoconstriction, okay? So as a result, what happens is if I decrease my sympathetic stimulation, okay, decrease my circulating epinephrine or whatever the case may be, what's going to happen is there's going to be less constriction of that afferent arteriole. Therefore, I increase the amount of inflow of blood from the afferent arteriole to the glomerulus, increases the glomerular filtration rate. Okay, going back to everything we talked about before. If for some reason there's a higher sympathetic stimulation, what it does is it constricts the afferent arteriole, which decreases the blood flow into the glomerulus, and that lowers the glomerular filtration rate. You ever notice that what happens is when you're active, when you're physically active, you don't have to go to the bathroom nearly as much. You don't. The body realizes you're probably going to be sweating, losing water, so what it does is decrease the glomerular filtration rate. How does it do that? Well, the activity also elevates sympathetic nervous system activity, which then causes a vasoconstriction of the afferent arteriole, decreasing blood supply to the glomerulus, therefore decreasing glomerular filtration rate, and we conserve the fluid. Logical. This is the beauty of it all, is things really physiologically make sense. You don't have to remember a whole lot, you just have to think about it, and everything sort of falls in place. And that's what the neural control really does in regards to the glomerular filtration rate. Okay, a couple things I should mention here. One thing's called a BUN. Okay, you might hear this working in the hospital. You might hear something called BUN. BUN stands for blood urea nitrogen. Okay, and again, I mentioned in my very first discussion on the urinary system that the way we get rid of the nitrogen waste is through the kidneys, and we always have a little bit of that nitrogen that's still left, in the body. Okay, we still always have a little bit of nitrogen. Okay, so when I take blood, just regular venous blood, okay, just like a regular blood test, one of the tests that they get is called a BUN, and BUN measures the amount of nitrogen in the body. Okay, what happens when the kidneys start to fail? What's going to happen? Logical, Logical. What happens is sounds like Mr. Spock here. It's very logical, Captain. Okay. What happens is logical. What it does, okay, is the urea or the nitrogen levels will start to increase. Now, urea is a nitrogenous compound, so they measure that level of urea in the blood as a nitrogenous compound. So if I see a patient and their BUN levels start to go up, what am I thinking? Their kidney function is going down. Why? Because they're not getting rid of the nitrogen, which they should. Okay. I use this some, and there are some limitations to that. There are a lot of other things that happen in the body that will change the BUN. Okay, do not say BUN, say BUN, blood urea nitrogen. If you say BUN, they're going to kick you out. Okay, there's a lot of things that influence the BUN. Okay, so I used to get it because everybody would always ask, well, what's their BUN? You know, and I'd say, oh, it's this and stuff like that. I always got plasma creatinine. Okay, plasma creatinine is a little bit more accurate than the BUN, okay? Because what happens is creatinine is also a byproduct or a waste product of protein metabolism and protein breakdown. And the only way the body gets rid of creatinine is through the kidneys, okay? So creatinine levels are directly related in relating kidney function. Okay, much more than sometimes some of these other things that interfere with BUN. Okay, also what I find is that plasma creatinine has a very low level. If I look at the BUN, the levels are usually between 10 and 20. If you look at plasma creatinine, it's like less than 2. Okay, and so what happens is small changes in the plasma creatinine mean more than the same size changes in the BUN, because the range is much greater and higher in the BUN as compared to the plasma creatinine. So personally, if I was looking at kidney function, or if I was looking at somebody who I was going to give an antibiotic for, and I was concerned about the high level of excretion through the kidneys and what it might do, before I called the nephrologist to see if I should adjust the dose of an antibiotic. A lot of times what I did is I got the plasma creatinine, okay? And it was a simple test, also just a venous blood test. You get it and it measures the level of creatinine. And if the creatinine levels are going up, okay, that's an ominous sign. Creatinine levels go up, it means the kidneys not get rid of it, it means the kidney function is going down. Okay. We also talked about the blood pH a lot in our last lecture, and what happens is we talked about how carbon dioxide forms with water to create H2CO3, which then naturally dissociates into a hydrogen ion, which is acidic, and a bicarbonate ion, which is basically basic. Okay, so what happens is, one way to control that, hydrogen ions reflect acid, bicarbonate reflects base. So therefore what happens is the kidneys can help to regulate blood pH by getting rid of too much. In other words, if I'm too acidic, they can take some of the more hydrogen ions and get rid of them in the urine. Okay, and the urine will become more acidic. Actually, when they test the urine, will become more acidic, or they could get rid of more bicarbonate, and that helps to control also acid-base balance. We talked to how the respiratory system is involved with that by taking the CO2 and making the H2CO3, blah blah blah. But also the kidney becomes involved in pH control, blood pH control, by getting rid of the extra hydrogen ion or getting rid of the rest or the bicarbonate that we don't need. Okay, to try to keep that pH within that 7.35, 7.45 range. Okay, hemodialysis. What happens if the kidneys start going bad? Okay, the toxins build up, nitrogenous products build up, toxins build up in the body, and these become very detrimental and can be lethal. They have to be cleared. Okay, since the kidneys aren't working, what are you going to do? You can't just go and say, well, let's just take out a liter of blood, because that doesn't do it, because we still have high levels of those toxins floating around. Okay, so what they do is they put a person on a hemodialysis machine. Okay, and this is done 3 times a week, and it usually takes multiple hours. It's not like going just get a shot and leave. We're talking about at the unit for multiple hours. Okay, and what they do is they usually put a little shunt in to make it easier to keep an area, a portal open. Then what they do is they'll stick the hemodialysis needle into the shunt, which is more of a synthetic material. Okay, and then what happens is it takes the blood out of the body, runs it through the dialyzer, which you see, and the guy sitting there reading the newspaper, runs it through the dialyzer, and the dialyzer does the same thing as the kidneys. It takes out the impurities, and then what it does, it takes the blood and puts it back in. So we're constantly taking something out, the materials out, and putting the blood back in, taking the materials out, putting the blood back in. So it's constantly has to go over and and over and over and over and over and over and over again. And that's why it takes a long time for the hemodialysis units. Now with the COVID, they're talking about some really problems in regards to running hemodialysis units, which are jam-packed. One thing I should say too is renal failure. The largest cause of renal failure in the United States is, sure, diabetes. Diabetes trashes the kidneys, okay? So if you have a patient that's a friend that's diabetic or a family member, try to get them under good control, because what happens is that diabetes trashes them. Eventually down the road, I have a friend who was from Chicago. He was he did a lot of stuff in regards to radiology. Okay, he looked at a lot of MRIs and stuff like that. And every so often, if I needed another opinion, I'd send to him. In fact, I was a co-author on an article about a foot mass, okay, in the leg. I did a biomechanical review of this mass, why it was there, or what it could be attributed to. And we needed somebody with that had more bona fides in regards to radiographic interpretation. And I called Bob, and Bob said, oh sure, he sent the stuff, send them the MRI images. He put his 3 cents into the paper, and we got published in a referee journal a number of years ago. Okay, but anyway, Bob, I used to be on a national committee for making national board, or actually not national board, board certification, which is above national board. Once you get your to be able to get your license, you go through the national board. But then when you go through a residency and you complete a residency, you become what's called board certified. And I was on a committee that we did the oral examinations for board certification for primary care and orthopedics, and there were 4 of us that did this in the country, and we'd meet about 4 times a year and make up all these oral examinations that we'd give to people who are outside practicing docs. They've been practicing for a number of years. They've submitted a number of cases to say that they have an experience, and then we question them and evaluate them, once a year in Chicago to do this. Anyway, Bob was in charge of the written exam, because a written exam to say that you actually take before you take the oral exam. And basically he got so sick and he couldn't come to the meetings anymore. And the reason why, he was diabetic and his kidneys were trashed. Okay, and basically he was on dialysis, and he went and got a kidney transplant, and the guy looked beautiful. I mean, he looked great, changed a whole lot. So anyway, dialysis is a hassle, but it's a necessity of life in those situations, simply because if you don't have it, the toxins build up and you'll die. Okay, it's a chronic care type of thing. There's also a bleeding risk that we get with hemodialysis, because when you take the blood out and you're running through the machine, you don't want the clot. So they actually have to anticoagulate the people, and then when they're done, they have to reverse that anticoagulation. Okay, so usually they give them heparin, and then when they're done, they give them protamine sulfate, which actually reverses the heparin effect, but they have to make sure everything is fine before they even send them home. Okay, now that's hemodialysis. On the bottom, you see something called peritoneal dialysis, and this can be done. It's not nearly as effective, and but it does allow people to stay at home and be a little bit more mobile. And in this situation on the right, there's a tube that looks like it's going into the peritoneal cavity, and the reason why it looks like that is because that's what it does. They surgically insert a tube into the peritoneal cavity, and then multiple times a day, this tube is left in the abdomen, coming out of the abdominal wall. You got to be careful sterilely, because what happens is that's one of the problems. If you take an infection and introduce it through this tube, now all of a sudden you have a peritonitis, which could be life-threatening. Okay, so anyway, what they do is multiple times a day they hang a bag with what's called a dialysate, or a solution that's going to actually draw out the impurities, or out the waste products. They take this and hang it, and then run it in, and they fill up the peritoneal cavity with this bag of fluid. Then they sit there for a while. What happens is then the peritoneal membrane, because it's very vascular on the inside wall, parietal peritoneum and visceral peritoneum, it actually allows that dialysate material to suck the waste products out through the blood through the peritoneal membrane. Then what they do is then they have to take and drain it all out. So after a while, when that peritoneal fluid is in there, then they attach another bag, which you see on the very bottom of the picture, and they drain it out. Okay, and as a result, that sort of works as well. It's not quite as monitored or effective as we find with hemodialysis, but it can be done at home. You can do it at home and other places, at the office and stuff like that. But again, there's a risk of infection and things like that with peritoneal dialysis. Best thing is probably in the end a kidney transplant, but there are so many people with kidney failure, and trying to find a tissue matched kidney becomes a problem. It's not like always easy to do. Okay. Urine, just a couple things about urine, okay? Urine, we know that when it finally gets outside the renal pelvis and down the ureter, it's changed. There's no more change. That's what it is. The only time it changes is if there's like an infection in the ureter or in the kidney itself, in the renal pelvis of the kidney, or in the bladder. Then all of a sudden it adds white cells, red cells, and stuff like that, okay? So there's no further change in the overall amount of urine. If I look at normal urine, okay, a couple characteristics of normal urine. We put out, like I say, about 2 to 3 liters, or 1 to 2 liters a day. Okay, it's usually a pale yellow to amber. Okay, you can see the change in the color of the urine. The more I drink, the more dilute things get. The clearer it looks, more closer to water. Okay, the less I drink and the more dehydrated I am, the more yellow it gets because there's more particulate matter. It's usually transparent. Okay, which means if I take and put something print behind it, I can actually look through the urine. I could read it from behind. So it should be transparent. If it's cloudy, it means there might be something floating around in there, like blood cells, a few blood cells, or white blood cells, and things like that might be floating around. Also, if you let urine sit, it starts to ferment. Okay, and actually you'll start to smell it. It smells pretty bad, and it starts to get cloudy and clumpy. It should be fresh. If it there's aroma to the urine, and the urine will change according to sometimes your diet. If you like asparagus, what happens is, I'm not saying to do this, but if you did smell your urine after you ate asparagus, it's going to have an asparagus smell to it. It's very strange. So some things you do eat will be reflected by the smell of your urine, okay? Again, it becomes ammonia when standing. Why? Ammonia is a nitrogen compound, NH3, okay? So the ammonia that's retained from the body is actually what we start to smell. Again, the pH is about 4.6 to about 8. The average is somewhere around 6. So it's sort of like in the middle. It's not acidic, it's not basic, and stuff like that. Specific gravity. Specific gravity means how much stuff is actually dissolved inside there. Okay, if there's not much dissolved in there, and there's very few things in there, it has a very low specific gravity. The specific gravity of water should be 1.000. So the specific gravity of urine is always going to have something in there. It's not going to be total water. So it could be anywhere from 1.001, which is actually really low, usually a little bit higher than that, all the way up to a concentrated urine of about 1.035. Once the urine gets really concentrated, all of a sudden my specific gravity gets higher. And that concentration is because there's a lot of particulate matter in there and frequently less urine. That's why your urine is darker when you're more dehydrated, because there's less water and more material that's dissolved in that, and therefore your specific gravity of the urine would normally be higher in those situations. Okay, and that's something to think about. Okay, a couple other things when they do a urinalysis, that a lot of times what they do is, for most of this, sometimes they just dipstick it. They have a little stick, okay, with little paper reagents on there, and they put it in the urine and then pull it out, and it causes a color change after a certain number of seconds. And they could look for things like protein in the urine. How much protein should I have in the urine? None, of course. Why? Because protein, like we mentioned in our previous discussions, is such a large molecule, it can't get through the tubules. It can't be, excuse me, it can't get through, it can't get outside the glomerulus, the glomerular capillaries. The glomerular capillary slits and holes are too small for the protein to get out. So I should find no protein in there. Glucose. Again, most of the glucose that's filtered is reabsorbed. We should find very minimal, if any, glucose in the urine. Where we seem to find a lot more glucose in the urine turns out in those situations when the blood glucose is so high that there's just so much to reabsorb, you can't reabsorb a lot, and some of it's what's called spilled into the urine. We look for red cells and white cells, which are abnormal. Okay, so I shouldn't see any protein in the urine. I should see no significant urine in there. I shouldn't see red cells or white cells. Red cells or white cells could be there either because of bleeding, such as a stone, a tumor, or something like that, or white cells because of infection. So they look for the urine for those things. Various microbes. In other words, they could take the urine, they do urine culture. They just see what grows. Okay. Ketones. We talked about this before. Diabetic patients, when they can't use glucose as an energy source because there's not enough insulin, or the insulin's not working, or it's not letting the glucose in the cell, because I need the insulin to get the glucose inside the cell. If the ketones are if I can't get the glucose in, we start to use fats and fatty acids for energy source, and the byproduct is ketones. Ketones are acids, and they're actually put out into the urine, and they show up in the urine. Okay. Also body pigments, myoglobin from muscle breakdown, which is significant, and also cell fragments, and that usually comes from the urinary tract. cell fragments are actually too big as well. Cell fragments are too big to get out the glomerulus. If there are cell fragments that are inside the urine, they're coming from somewhere in that urinary tract, the bladder, the ureter, the renal pelvis, or something like that. We also find what are called casts. Now, I mentioned casts a little bit before. What happens is if I do have white cells that get in the tubules, which sometimes happens, if somebody has an infection, and what happens, it causes the wall of the glomerular capillaries to become excessively leaky. Okay, and white cells, which are huge, somehow get out, and we get white cells that will actually form inside the tubules. Or if there's some other way white cells get inside the tubules, they become sluggish. They sort of like they're too big. It's sort of like everything else wants to float down the stream, but the white cells can't. They're just too big. They just float slower. Okay, so they start to congregate, and they actually create a cast or a mold of the tubules. They finally get pushed out through the collecting duct and end up in the urine. So when they look at the urine, they'll actually find these like clumps of white cells that are clumped together, and they're called white cell casts, or sometimes they call hyaline casts, which is basically because protein leaking out. So those are things there. We could also see stones, and the term for stones or stone particles, which is basically called calculi. Calculus means stone, and so we may find parts of that, or sludge or sediment. Sometimes you don't find a stone, but you find the material that leads to a stone, like calcium oxalate, sort of like in a gravel inside the urine. Okay. And oh, by the way, one last thing I want to mention before I let you go is the kidneys are also involved in red blood cell production. I briefly talked about this before, and what happens is if I have anemia, a low number of circulating red blood cells in the body, the kidney detects this. The kidney then secretes a hormone called erythropoietin. Erythro means red. Poieto means formation. So what happens is erythropoietin is made by the kidney and put into the circulation. The erythropoietin goes to the area of the bone marrow, and in the bone marrow, it tells the stem cells, which are about 1 to 2% of all the cells in the bone marrow, tells them, hey, I need you to make red blood cells, because that stem cell could make a red blood cell, white blood cell, platelets, whatever. Okay, so it tells the stem cell that they need more red blood cells. Okay, so it stimulates the stem cells in the bone marrow to make red blood cells, and therefore what happens, the red blood cells are produced, put out, and stuff like that. So that's one of the things I think you should know. It's called erythropoietin, or sometimes it's called EPO for erythropoietin. And so the kidneys are really responsible in some way of maintaining that red blood cell concentration, that red blood cell percentage or numbers that we see in our normal typical circulation. And if not, if anemia occurs and the red blood cell count goes down, the kidneys detect it, secrete that erythropoietin. The erythropoietin goes to the bone marrow, tells the stem cells make more red blood cells are made. Okay, tells you sort of like PS, sort of tells you why people with kidney failure may frequently have anemia. Why? Because if the kidneys aren't working, guess what they don't make also? They don't make erythropoietin. So as a result, the cells aren't working, and the kidney is basically a lump of garbage that sits back there in the flank. Then what happens is they don't make the erythropoietin. Okay, and as a result, it's common. It's called anemia of chronic disease. Okay, or anemia of kidney failure, because what happens is there's no erythropoietin to stimulate the bone marrow stem cells to make red blood cells. And that sort of gets me to the end of the urinary system. Hopefully, it's a simple in regards to organ. Okay, we got 2 kidneys, 2 ureters, a bladder, and a urethra. My gosh, I mean, how much simpler could it be until we start to think about the physiology, the filtration, the reabsorption, the secretion, the control of the glomerular filtration rate. With the juxtaglomerular apparatus and with the afferent arterial, with the efferent arterial, what happens in the tubules, it becomes very complicated. However, I think it's sort of something that's really interesting. It's really sort of neat how it works. But you know what? I think everything in the body is sort of neat how it works, and that's why I like this physiology stuff, okay? But anyway, hopefully this gave you a little bit of idea how the kidneys work a little bit more than what you had before. And like I say, if you ever have any questions, please, by all means, give me a call. I'll be more than happy to answer any questions that you might have. So we'll talk to you later. Bye-bye. Be safe and be healthy.